Hey everybody, welcome to V34. In this video, we're going to look at the movement of chromosomes between cells. And this picture truck, just a little way of reminding us what we're doing in this video. Cells need chromosomes. Why? Before we talk about how DNA gets moved around or how chromosomes get moved, let's talk about why. Recall that DNA is an instruction set. A chromosome is a large, twisted up version of DNA, and we have 46 of these per cell. So when we see a chromosome here, this whole thing is a chromosome. If you could look very, very, very carefully at it, you see it's all DNA twisted up together. Segments of that DNA, remember, are called genes. So the genes spell out proteins. So a long way of getting to this idea that a chromosome really is like a book. It's got a huge set of instructions on it. Individual pages are going to be individual instructions for making something. Kind of like a recipe book. That's a good analogy. So cells need chromosomes. Therefore, we're going to have to make sure they get them. We have here in our example, we have a purple one and a green one here. We have two chromosomes, and recall this term we used in class. They are homologous. Homologous means they have the same kinds of information on them. If I could go back and again locate the genes, there might be a gene here and a gene here. We'll call this one for right now, we'll call this the version that you get from dad. Dad. It's hard to write with a mouse. And mom. So you got a trait as, let's say, again, this is going to be for ears. So this is the ear gene, and maybe down here we have a gene for making an enzyme for digestion. So what's called a D gene. So you may have gotten the same versions of these from your parents. You may have gotten different versions. The point is, a gene is a set of information, and we need to make sure that all cells that come from one cell, all succeeding cells have all the same information that they need so they can make proteins too. So the first step in the process, if you will, is to copy everything. So we have this, of course, drawing a line here, represents the copy of this one, and this represents the copy of that one. So now we still just have two chromosomes, but we have copies of them, and we call the copies of a chromosome chromatids. And they're joined by, there's the word, they're joined by a centromere. So cells are going to copy their DNA. So again, it can be confusing to think of this like this is still just one chromosome, but this is one chromosome attached to its copy, one chromosome attached to its copy. There's still just two chromosomes here, but we also have some copies as well. So the task is going to be, when we create new cells, we need to make sure that a cell gets a purple copy and a green copy in one cell, and a purple copy and a green copy in the other one, so they will be exactly alike. So let's get an idea how this works. This chart here represents the cell cycle. Think of the cell cycle like the cell's lifetime, if you will. If you wanted to start here just as a place to begin, this is when a cell is born. This is kind of its birthday. It's going to go through a series of growth, of time of copying its DNA, some more growth, and then the actual division process. And the division, there it is up here, is mitosis. So a cell is produced, grows, copies its DNA, grows some more, and divides into two daughter cells. Each of these cells will then grow, copy its DNA, grow some more, divide, and produce two new daughter cells, and so on, and so on, and so on. I am not going to stress you out by making you look at mitosis in great detail. I'm far more concerned that you understand that mitosis is a way that we separate DNA into two daughter cells. You may find it useful to look at the terms prophase, metaphase, anaphase, and telophase as just helping you see what you're looking at on this chart, but I'm not going to ask you to give me much detail about all the processes that happen in prophase versus metaphase. I do want you to see that a cell goes through a cycle of growth, copying its DNA, more growth, and then division. Let's look at this for just a second. This reminds me of the chart we just saw when we had a chromosome, and then during the copying phase, we're going to end up with two sister chromatids for each chromosome. So when we say DNA copying, that's what we mean. That's when we produce sister chromatids. Now, this process is carefully regulated. Let me come over here and highlight it some more. And let me do here, oh, here's a different color. Highlight it some more. This is a regulated process. Sorry. This is a regulated process. It's got to be. We have to carefully control how often cells divide and then grow and then divide and then grow. And you'll see what happens in a future video when it's not well regulated. So let's move on to just, and again, I'm not going to hold you responsible for all the details, but just so you get an idea of what's happening. In interphase, that's when the cell when it's growing and copying its DNA. When we actually start to divide, where's my mouse? When we start to divide, you see the chromosomes here, and they look like little twos kind of strung together because we recognize that's a chromosome with its two sister chromatids. Eventually, we're going to attach some wires or proteins, better said, to each of these pairs. So there's one stuck on one side and one stuck on the other, and that's happening during prophase. We will get to a point where all of the chromosomes are lined up in the center of the cell, and they're all attached, and I'll give you this term, they're all attached to the spindle, which is just a series of proteins that attach to each side. And the way you can see that a little easier, let me just get rid of this. If you come over to this slide, just imagine that here we have one sister chromatid of a chromosome. Here is the other one. Notice how they're exactly copies of each other. Exact copies. Yep. And you can imagine that this represents the spindle, where it's attached to one sister chromatid, and this is attached to the other. What happens during mitosis is we separate the sister chromatids because they actually represent identical copies of the chromosome. So this happens in anaphase. You see now how we're separating this one from its sister, this one from its sister, and eventually these are going to end up on the side of this cell, and on the other side of the cell they're going to end up going over here. Eventually they're going to come on each side, and we're going to pinch off and form two. Let me move out of the way here. We're going to form two new daughter cells that are exactly genetically alike. They have the exact same number of chromosomes because we split up the pairs. We split up the sister chromatids. So when we look at the result of mitosis, we see this. The result of mitosis is that we've produced two, that's important, two daughter cells. Two cells, yes, that are genetically exactly the same. How do I know this? Because the DNA was copied exactly, and I put one copy to each new daughter cell. And therefore, this should be obvious. We have the same number of chromosomes as we started with. Recall in humans that we have 46 chromosomes. We would expect two daughter cells that are produced, each of which is going to end up with 46 chromosomes. Now, this goes back again to our conversation about the control of the cell cycle. This diagram is showing you the same thing you saw before, just in a little different way. This blue part here represents where the cell actually divides. The G represents growth. The part where I'm covering up is where it copies itself, copies the DNA rather. Here's where there's more growth, and then there is cell division. I'll come back to this theme one more time. It is carefully controlled. How is it controlled? Once a cell is formed, like you're seeing here, and it goes through its growth phases, there's going to be a series of checks to make sure that cell is prepared to continue its life. This one is called a G1 checkpoint. I'm not concerned that you know the names of it, but at this point, the cell is evaluated. And if the cell has grown enough, it has gone through enough growth phases or enough growth to actually get large enough, it passes the test and is allowed to continue to this phase, where, as you remember, this is where the DNA is copied. It will continue along the cell cycle, and then there's going to be another evaluation point where the cell checks itself. Did it copy all the DNA correctly? Is everything in place? Did everything go okay? If it does, if it passes that test, it goes on to mitosis, and there's yet another checkpoint. We will evaluate whether the spindle formed correctly, whether the chromosomes were moved properly, and if so, the cell is allowed to divide, and now we have 2 new daughter cells that will continue the cycle. So at critical points in the cycle, that cell is going to be evaluated to determine whether or not it meets the quality standards to continue. If it's all done well, cells that are messed up are killed. They're not allowed to continue, or they're stopped, and we won't get into that part too much. But cells are only allowed to continue if they're healthy. They're only allowed to divide if they're healthy, if this whole cycle is working correctly. So this is the cell cycle, and we'll see in the next video what happens when the cell cycle control isn't proper and isn't working. That's it for video V34.